Hi everyone, so this is the Bio 25 final exam review, and this is the first review of the 4 sessions total. And yeah, so this slide is just the intro slide, and yeah, there are 3 other review sessions. And this is just a reminder not to rely too heavy on these review sessions as a way to prepare for the exam, because we've only picked out the most important stuff from each week. But there's definitely more details that you will see in your own notes that you should go back and revise. And another thing I wanted to say was thank you for filling out the poll, because it was helpful in determining whether or not we were going to do these sessions. So yeah, that's just the poll. And sorry, Rachel, do you want to go ahead with this section?
Yeah, so, wait, I want to just turn that sound off. It's gonna bother me.
I think only you can hear it. I don't think everyone can hear.
Oh, okay, it is going to bother me. Okay, well, I guess I can't change it. Okay, hi everybody. Hope your break is going somewhat well. I know it's obviously hard being here and stuff with everything going on. We've all finals, but we'll try to make this quick, but also we'll spend time on things you guys want to go over more. Will the PowerPoint be sent out? I don't know, Michelle, were you planning on sending it out?
It will be sent out at the end of the session with the video on YouTube.
Yeah. So we'll send out the recording of it, so you can watch the recording to go through it. Okay. All right. So we'll start with amino acids, because that's how we started the class, and you kind of need to understand these before you can understand everything else. So amino acid structure. This is the basic structure. If you remember from earlier in class, you have your amino group, which is the NH2, your carboxyl group, and your alpha carbon connected to a hydrogen and an R group. So the R group is, remember, the variable part of the amino acid. This is what characterizes your amino acid. This is what gives it its properties, whether or not it's polar, nonpolar, acid base. And that's so this is the backbone, the amino group, the alpha carbon, and the carboxyl group. And the R group sticks out from the backbone, so does the hydrogen. You don't need to know the organic chemistry of the positioning and everything, but that's just the basic structure that all amino acids share. Amino acids are pH dependent. They're dependent on the pH, depending on their state, whether or not they're positively charged, negatively charged, or neutral. So a zwitterion is a neutral amino acid. That means that it has the positive charge on its amino group and a negative charge on its carboxyl group. To make the whole thing neutral. Here is just an example, as you can see on the diagram, is glycine, which you don't need to know that that is glycine, but its R group is also neutral, so that makes the whole thing neutral. If you had a charged R group, like I don't know, aspartic acid or glutamic acid or something, then it might be a different pH to make it neutral. But as long as your pH is neutral, your amino acid will be a zwitterion. But that's not that important for your exam. I also just quickly want to say that there are 20 amino acids. There's 11 polar. 2 of the polar are acidic, 3 are basic, 6 are neutral. There are 9 nonpolar. At physiological pH, which is 7.4, which would be in the cell, acidic amino acids are negatively charged, and basic amino acids are positively charged. Nonpolar amino acids are hydrophobic, and polar amino acids are hydrophilic. So the formation of the peptide bond, which is the bond between your amino acids to make your primary structure, it's a dehydration reaction, which means water is formed as a product. It's a covalent bond, which is very strong. We're going to get into structures of proteins, but just a reminder that this is the only structure. This is one of the only structures that has this covalent bond. Breaking of a peptide bond is a hydrolysis reaction, which means it requires water to be put in and releases energy when doing so. the peptide bond it's very strong. That's one thing you really need to know. And remember, just for a side note, that the amino acids are read from the N terminus to the C terminus. That's important when you're looking at these diagrams.
Okay, so now I'm going to go over levels of protein structure fairly quickly because I wouldn't expect more than like 1 or 2 questions on the exam about this. But let's get started with primary. So primary structure of proteins is basically just the sequence of the amino acids. So they're held together by peptide bonds, which are strong covalent bonds like Rachel was just talking about. You can only break these by like enzymatic reactions or things with like lots of energy. Important to know that just the important thing here is that the peptide bonds that hold primary structure together are covalent. And again, it's just the sequence of amino acids. So secondary structure is the interactions between the backbone. The 2 types of secondary structure are the alpha helices and the beta sheets. She didn't talk too much about the difference between alpha helices and beta sheets this year, but basically alpha helices leave the R groups on the outside, and in beta sheets, R groups alternate up and down in intersperse. The important distinction here is that in secondary structure, hydrogen bonds hold secondary structure together. Hydrogen bonds, as you know, are non-covalent, so they're weaker than covalent bonds, and which means that it's easier to break a secondary structure than a primary structure, for example. The next level of protein structure is tertiary structure. So tertiary structure is pretty much just like one peptide together. So this is as a result of interaction between its R groups that give it an overall protein shape. These could be various types of interactions between R groups, like hydrophobic interactions, ionic interactions, hydrogen bonds, disulfide bridges. If you remember, disulfide bridges are interactions between 2 cysteine amino acids, and that forms a disulfide bridge, and that's a very strong bond that can happen in tertiary and quaternary. And next we have quaternary. So quaternary is has the same interactions as tertiary, but in quaternary, it's when more than 1 subunits are interacting between each other. And these are, again, interactions between backbone and R groups. So quaternary is basically a bunch of subunits trying to interact together to give the protein its overall shape. Again, subunit is a subsection of a giant protein, and we'll talk in a second about the difference between subunits in a domain. One example is hemoglobin, which has multiple subunits, and this is one example. So like the blue is one subunit, the green is another subunit, etc. Not every protein will have a quaternary structure. It could just be a protein with one subunit, so it'll just have a tertiary structure. But I'd say most proteins do have a quaternary structure. So one example question, if you want to answer this in the chat or unmute yourself real quick. What effect would a molecule that disrupts hydrogen bonding, such as urea, have on protein structure?
The protein part?
What did you say? Sorry.
I said that the structure would fall apart.
Well, yes, but specifically which part of the structure? Like
be everything except primary, right?
So I would say that urea would disrupt the secondary structure because that is the thing that's held together by well, you're right. It wouldn't disrupt primary structure because that's held by covalent, but it would. I guess what I was looking for was it would primarily disrupt secondary because it unfolds the alpha helices and beta sheets. But yes, you're right. The primary would still be intact.
Does anyone have questions so far on anything you've gone over? You can put it in the chat or unmute yourself.
Hydrogen bonds are also involved in stabilizing tertiary structure, right?
Yes, but I would say they're more prevalent in secondary structure, but yes, there are some hydrogen bonds involved in tertiary and quaternary structure.
Okay, thank you.
So just to recap, tertiary structure is a single polypeptide chain and how it folds. Quaternary structure is multiple polypeptides coming together to form a three-dimensional shape. And these are both stabilized by covalent and non-covalent R group interactions, the covalent being the disulfide bridges, and the non-covalent being things like hydrogen bonds, electrostatic interactions, van der Waals, ionic, stuff like that. Okay. And lastly, for this section, go over the difference between domain and a subunit. So a domain is basically just a section of a protein that folds independently from the rest of it. There could be multiple domains within a subunit. There could be multiple domains throughout the protein, but basically it's just a section of a protein that folds independently from the rest of it. And it usually has a specific function. So there could be one protein domain that allows a protein to bind to a receptor, another protein to activate another domain to activate a molecule within the proteins or within the cell or something like that. So domains are just a section of the protein that fold independently from the rest, and they have their own function. These are usually evolutionarily conserved. So like a domain from an alligator will be similar to a domain on a human or something like that. That's basically what it means by evolutionarily conserved. And like I was been saying, a subunit is just a part of a protein that come together, or just like a single polypeptide chain. And multiple polypeptide chains can come together to form quaternary structure. Quick question, are there any subunits in tertiary structure?
I think
Repeat
so.
the question. Are there any subunits in tertiary structure? I see one yes and a bunch of no's. So I would say that there is one subunit in tertiary structure. So a little bit of a trick question, but so tertiary structure is just a single subunit, and then quaternary structure would be multiple subunits. Any questions on that? Otherwise, we're going to move on to our group interactions.
Yes, are there any questions? Oh, sorry, go ahead.
No, sorry, you can go.
No, I was just gonna ask if there was any questions, but you can go ahead and ask your question.
Okay, I have one question. So like, are you saying that I thought subunits have to be 2 or more polypeptides. So how would it be possible that the tertiary structure has a subunit or is a subunit, I guess?
It says the quaternary structure is the result of 2 or more polypeptides. So this isn't the direct definition for a subunit, I'm sorry. So quaternary structure results from 2 or more polypeptides, but a single polypeptide chain is a subunit.
Okay, thank you.
Yeah, generally for this class, Professor Francis uses subunits, like plural subunits, to describe the components of quaternary structure. So yeah,
okay,
when you have 2 or more subunits, then you have quaternary structure. So just like the hemoglobin is like a giant protein composed of like smaller subsections or subunits, like the different heme groups, which were highlighted in different colors on the other slide. So hopefully that helps.
Yeah, thank you.
Okay. Cool, so we'll just keep going then.
Okay, so our group characteristics, interaction and bonds, this was something that I know a lot of people had a little trouble with on the first exam, but I'm really not sure how many questions Professor Francis is going to ask about it, but we'll go through it. So just quick recap, polar versus nonpolar. Polar is the uneven distribution of electrons. So this example of water. Water is a very polar molecule. Here you have these partial positive charges on the hydrogens and the partial negative charge on the oxygen. The oxygen is much more electronegative than hydrogen, so it pulls away more electrons. So that's why there's an uneven distribution of them. Whereas you have your nonpolar molecules, which are electrons that are evenly distributed. It's usually a symmetrical molecule, and the same charge will be across the molecule. So here you have on the nonpolar side, carbon's partially positive and your chloride is partially negative. But since this is a symmetrical molecule, the charge is evenly distributed and there's no pusher pull of electrons throughout the molecule. So it's just a quick recap on polar and nonpolar. So when we talk about amino acids, there's certain categories of R groups for your amino acids. Polar charged R groups are down here in this blue box. It's the green and the purple box are your polar charged R groups. The blue box is your polar uncharged. So your acidic amino acids are here in this pink purple box. That's your aspartic acid, glutamic acid, and then your basic amino acids are in green. And then these blue amino acids are all polar. So that, like we just talked about, uneven distribution of charge. And then yellow are all the rest are nonpolar. So your polar charged R groups, which would be your both basic and your acidic, they'll usually make ionic bonds. They'll make electrostatic interactions. Your polar uncharged R groups here in blue, they'll usually make hydrogen bonds. They have the ability to make the hydrogen bonds because they're very hydrophilic. So they have a lot of carbon and oxygen bonds, carbon, nitrogen, oxygen, hydrogen, nitrogen, covalent bonds. Because remember, to make a hydrogen bond, you need an oxygen, a nitrogen, or a fluorine, and your hydrogen, obviously. But when we're talking about cellular biology, we usually don't need to think about fluorine because you're usually not going to have it. So as long as you have your nitrogen and your oxygen and your hydrogen, you can usually make a hydrogen bond. And these polar amino acids are pretty good at that. They're hydrophilic and they have a large difference in electronegativity. And then your non-polar R groups up in this yellow box, they're hydrophobic. So you'll, sorry, they have a lot of carbon-carbon and carbon-hydrogen covalent bonds within their structure. So this is the example of the hydrophobic effect. So just a quick question, where would you usually find your nonpolar amino acids within the structure of a protein that's folded? Where would you usually find them? What's that like hydrophobic effect?
Did you say where are we going to find the nonpolar sections on the protein?
Yeah.
On the inside?
Yep.
Away from.
Interior, yeah, exactly. So that's what the hydrophobic effect means. You'll find the hydrophobic amino acids on the inside of the protein away from the aqueous solution because they don't want to be near the water or the polar solution. Meanwhile, you'll find your polar and your charged amino acids more likely on the exterior of your protein. They also, just quick, that these nonpolar amino acids have similar electronegativity.
I have a quick question.
Yeah.
What is it called when the positive charge and the negative charge interacts? Like, is there a name for that interaction?
When positive and negative interact, I'm pretty sure Professor Francis would just want you to know that that's electrostatic interaction or an ionic bond. I don't think she'll want you to know the actual names because I doubt she'll ask you that. But yeah, it's just the electrostatic interaction.
Okay, and also, what is the interaction called between a nonpolar and polar?
Nonpolar and polar, it's either induced dipole. Hold on, let me pull up my notes because I might have it. Jason, do you remember? I think it's induced dipole. I could be wrong.
Yeah,
though.
I think it's just induced dipole, but she just refers to it as like dipole interactions.
It's just a dipole. That's what I thought, but
Yeah.
I know yeah. So yeah, a nonpolar and polar would be a dipole because you have a uneven distribution of charge going towards the polar amino acid. But yeah, that would be the simplest answer, I think.
Okay, thank you.
Yeah. Does anyone have any other questions before we go on to other topics? So steric hindrance is basically just the bulkiness and repulsion based on the R group size. So the R group size can affect the structure of the protein. Their size and their chemistry matters. So we just talked a little bit about their chemistry, the hydrophobic effect where they'd end up on the protein, but their size also affects the protein and folding, because if you have a big bulky amino acid, kind of like phenylalanine, there's going to be a high steric strain, which would make the backbone more rigid. So it is aromatic. I'm just going to start with phenylalanine because I was talking about it. Just move backwards. But so phenylalanine is aromatic, which means that it's very stable, but it's very big. So it will cause that high steric strain. And then if we move on to protein, it's cyclic, so it's not as stable as aromatic, but it can form a helix, but it would make the backbone also more rigid and cause high steric strain because it's big and bulky. And proline's the only amino acid where its R group is connected to the amino group, which means that there's more steric strain. It will never be located in the middle of a polypeptide. There's some other things about proline too. You won't find them in certain places, but you guys don't need to know about that. And then if we move to glycine, it has a very small R group, it's just a hydrogen. So there's little steric strain, which will make the polypeptide more flexible in the backbone, which is kind of important when you're, folding and turning the protein to make bonds. You need that backbone. You're going to want that backbone to be flexible in order to like fold in on itself. You wouldn't want it to be rigid.
Oh, does anyone have questions on this slide? No? Okay.
Okay, so now we get into the interactions between each structure. So like we said before, primary structure is the sequence of amino acids. It's held together by covalent bonds. These are peptide bonds. To form a peptide bond, like I said before, is dehydration synthesis. Water is released, as you can see in this first diagram. That you lose this OH and the H to make H2O. Your secondary structure is the formation of the alpha helices or beta sheets, which is caused by hydrogen bonding between amino groups and carboxylic acid groups. So just a quick recap, hydrogen bonds are non-covalent bonds. I know they can be kind of confusing at times, but if you can see here on the diagram, you can see this line going up between the two like ribbon part of the alpha helix. That's where your hydrogen bonds are. So it's stabilizing between the the turns for the alpha helix, and then for the beta sheet, you can also see the lines in the middle. It's stabilizing the beta sheets by holding them together. So the hydrogen bonds, I think we had a slide on it on the first exam review slide about what hydrogen bonds are. So if you ever need to look back at them. But so basically the first molecule has to have a less electronegative atom with either an oxygen or a nitrogen. And the second molecule, the hydrogen needs to be bound to an oxygen or a nitrogen. So the important takeaway for hydrogen bonds real quick is that the oxygen or the nitrogen need to have a negative dipole, and the hydrogen needs to have a positive dipole on the other molecule. So if you look here, you can see that this hydrogen is connected to another hydrogen, or actually a better example is one of them is this hydrogen's connected to oxygen to make a hydrogen bond, because that oxygen has a negative dipole connected to a carbon, which is less electronegative than oxygen. And then the hydrogen has a positive dipole because it's connected to nitrogen, which has a higher electronegativity than hydrogen. So that's just quick on hydrogen bonds. I don't think she'll really be asking you specifics about them on this exam, but just in case. Secondary structure, does not involve the R groups. This is pretty important. It's only the backbones for your bonding. And then your tertiary and quaternary structure. So this is the whole polar charged versus polar uncharged. Polar charged R groups can form ionic bonds with each other, so either two acidic or two basic, or an acidic and a basic amino acid. or electrostatic interactions with polar uncharged. So if you had like serine and lysine, so that blue box with either that green or pink box would make electrostatic interactions. Just quick note that they form bonds with charged molecules of opposite charge. And then we have polar uncharged R groups can make hydrogen bonds with other R groups or the backbone. This is only true for your tertiary and quaternary structures. R groups are not involved with hydrogen bonds in the secondary structures. So when you hear hydrogen bond in secondary structures, that's just between the backbone, not with the R groups. And then just the nonpolar interactions you'll find in tertiary and quaternary structure are van der Waals interactions. That's just between two nonpolar R groups. And hydrophobic R groups are typically found inside the protein, which is the hydrophobic effect. Does anyone have any questions so far? No? Cool. Okay. So we briefly mentioned disulfide bond when we were talking about structure, but just to quickly go over it, covalent, it's a covalent bond that only occurs between two cysteine molecules, and it's only in tertiary or quaternary structure, so you will never find it in secondary or primary. It's only tertiary and quaternary. It stabilizes structures very well because it's a very strong bond. And then this is just a quick diagram of how it's formed with oxidation reduction reaction, which you don't really need to know, but just remember that it's between two cysteines with their R groups are these SH groups. They're thiols, I think, the organic chemistry name, but anyways, you just need to know that they're between two cysteine molecules, and they're only found in tertiary and quaternary structures, and they stabilize it very well. So quick bond summary, because I know that was a lot. Your covalent bonds are your peptide bonds, which you find in primary structure, and your disulfide bonds you find in tertiary structure. Your non-covalent bonds are your ionic bonds, so that's between polar charged amino acids, hydrogen bonds between polar uncharged, and van der Waal interactions between nonpolar. And then we have this little chart. So primary structure, you have your peptide bonds and backbone interactions. Secondary structure, you have hydrogen bonds, which are only in your backbone interactions. No R groups here. And then tertiary and quaternary are very similar and have very similar structure or very similar bonding. So they both have covalent and non-covalent bonds, and they both have side chain and backbone interactions.
Are there any questions? No questions?
No, I think you're good.
Someone's mouth. All right, I'm just gonna keep going. So this next part is protein unfolding and misfolding. And the main ideas for this is that in the environment that a protein resides in can impact the folding and function. And there are a bunch of environmental factors we discussed, and some of these include temperature, light energy, and then pH and salt concentrations. And a cell has the ability to regulate environmental factors like temperature, which is also thermal energy, and then salt concentrations, because they can have an impact on non-covalent bonds. And then we defined a misfolded protein in this class as abnormal, and it doesn't function properly. And when a protein unfolds, this is called denaturing. And then in a similar fashion, when a protein refolds, it's called renaturing. And then having a protein that is misfolded or unfolding can be harmful to the cell. And cells have responses that cells have evolved responses to fix these. Does anyone have questions so far? Okay. And so main idea for this portion is anything can have the potential to disrupt the non-covalent bonds of a protein. And this is because for this class, non-covalent bonds are weaker than covalent bonds. And I know most of you probably have taken Gen Chem or maybe other chemistry courses, and you've learned that ionic is stronger than covalent, and that might sort of be difficult to grapple with. But just know that for this class, we are considering the covalent bonds to be the most strongest bonds. And so that's why for this part that the non-covalent bonds are significantly weaker. So we'd expect that anything like temperature or light energy can disrupt these bonds. And so these weaker non-covalent bonds are van der Waals, electrostatic interactions, which are called ionic bonds, and then hydrogen bonds. And covalent bonds could be disrupted, but this requires a super, super strong intensified factor. So that means very, very high heat or a light source. And so this is why for the majority of the time, when you're asked about environmental factors for this class, just assume that the covalent bonds are strong enough to remain intact. And these covalent bonds are peptide bonds and disulfide bridges. Does anybody have any questions on this slide? No? Okay, I'm gonna keep going. And so these slides are just gonna break down a few of the environmental factors. So the first of these factors is controlled by the cell is thermal energy, also known as heat. And heat can attack the non-covalent bonds and cause denaturing to occur. And an example of this is prevalent when you make eggs. So when you cook scrambled eggs, the egg will coil and shrivel up into a clump because the hydrophobic core is broken and nonpolar amino acids sort of scramble around and find each other. And extremely high temperatures can affect covalent bonds, but like I said earlier, we're just going to focus on the non-covalent bonds in this class. And Professor Francis mentions the Goldilocks hypothesis, which sort of says that in general, when you have more thermal energy, you have more stable bonds, but however, more stable is not always better for a particular protein. So even if the temperature is higher or lower, it doesn't mean that the protein will function better or worse. And for humans, our cells have evolved mechanisms that work for them to function best. So for us, we work best, the cells and proteins in our systems work best at 37 degrees Celsius.
I have a question.
Yep.
Wouldn't less thermal energy mean more stable bonds? Because like increased heat would make them less stable?
Actually, I think that might be a typo in there, but I'll need to check my notes because I think I may have also had that written down for the other review, but I'll check my notes at the end of this and I'll send an email if it's wrong, but I think you could be right on that. Jason or Rachel, do you have any thoughts on that?
Yeah, I believe that more thermal energy would make them more unstable.
But yeah, I'm just gonna I'll check my notes because I wrote it based off notes I had for the class, so there could be potential that it's wrong. But if I find that it's wrong, I'll definitely send an email correcting it because I don't want people to misinterpret that.
Okay, thanks.
So yeah, so I guess what I meant to say for Goldilocks hypothesis is even if your temperature is higher or lower, it doesn't necessarily indicate that the protein can function better or worse. Yeah. Okay, so are there any more questions on this slide? Okay. And for pH and salt concentrations, these can affect charge interactions, which are the electrostatic ionic bonds. And cells have a certain pH that they work best at, and it allows them to control the amount of salts that are around. And that's for pH and salt concentration. And then the last environmental factor I think I have, that this is something that's not controlled by the cell, which is high energy wavelengths or light. And light is induced energy into a system, and this can disrupt non-covalent bonds that hold proteins together. And just like we said for the heat, if you have extremely high light intensity, that can have the ability to break covalent bonds, but you won't be asked about that for this class. You just have to assume that when you have certain environmental factors that the covalent bonds are strong enough to remain intact, and then it would only disrupt the non-covalent. Unless she was to add something else that would disrupt the covalent bonds. But yeah, does anyone have questions on that? But yeah, I just remember that I think there was either a question on the exam or one of the problem sets that she added different factors like detergent and like reducing agent. But depending on what your factors are, if they're outside of the environmental factors, I think they have the ability to affect the covalent bonds. So like if you just had heat and light, it wouldn't be enough to break the covalent bonds. But then I think she either added, I think it was either detergent or reducing agent. I just can't remember which one it was, but it was able to affect the covalent bonds, I believe. But yeah, does anyone have questions on this slide?
So did heat only affect non-covalent bonds, technically, apart from like super high temperatures?
Yeah, so heat only has the ability to affect non-covalent bonds. That's correct. Okay, so any other questions on this part? Okay. Oh, I thought I skipped over. Yeah, okay, so now I'm gonna move it on to how to resolve a misfolding protein. And so some important points is that cells have robust mechanisms that respond to the problems of protein misfolding. And there are two responses we spoke about. So in the cytosol or nucleus, there's a heat shock response, which involves sending proteins to the proteasome in a stress response pathway. And then the other response we talked about was the unfolded protein response, or the UPR. And this occurs for proteins that go to the endomembrane system. So this is an ER-specific stress response. And a lot of neurodegenerative diseases are caused by misfolding proteins or the formation of aggregates. And I don't think she spoke about this in class, but I just thought it was a cool thing to include. But the mad cow disease or Creutzfeldt-Jakob disease is caused by clumping of prions, which are just like misfolded proteins that don't behave correctly in the brain. And then when proteins aggregate and form clumps, they become insoluble because the protein unravels and the hydrophobic core becomes exposed. And does anyone else have questions on this slide? Okay, I'll just keep going. And for these response mechanisms, there are these things called chaperone proteins, which are recruited to help other proteins fold or stabilize them when they're unfolding or misfolding. I believe chaperone proteins are involved in the UPR response. And they are able to sense misfolding or unfolding. And the way I remember about what these proteins do is I just think of them as chaperones on the field trip. And the purpose of chaperones on the field trip is they're just parents that volunteer to supervise students and to make sure that they're behaving and they're not out of control. So it's sort of similar for in the cell when you're looking at the UPR response, because chaperone proteins help misfolded proteins fold correctly or behave properly. Does anyone have questions on this slide? Okay. Okay, so the first response was heat shock response, and this basically all that happens here is that you have a protein that's misfolded or not folding properly, and it gets sent to the proteasome where it eventually gets degraded. And Professor Francis likes to think of the proteasome as a garbage disposal, kind of like the one you have in your sink where you just put stuff there and then it grinds it up and destroys it. So that's pretty much the same thing what happens to a protein. And during this response, you have the upregulation of chaperone protein production. And this response occurs in the cytosol and nucleus. And this image just sort of shows the idea with the proteasome. I think the bottom of it got cut off, but basically there's just the good behaving protein on the top is able to go on and promote cell survival. And then the bad protein at the bottom, which I tried to make look different, has something wrong with it. So then it gets, I think it gets tagged with ubiquitin, I believe, and then it goes to the proteasome. But all you need to know for this one is that cells figure out that something is wrong with a protein that's misfolded in the cytosol nucleus, and then it just sends them to the proteasome where it gets destroyed. So that's just the main idea for this slide. Does anyone have questions on this slide? Okay, I'll keep going. And the second response we spoke about in class was the unfolded protein response, the UPR. And this response occurs in the ER. So it's only for proteins that go into the endomembrane system. And basically what happens here is that you have cells that detect unfolded proteins in the ER, and a few events happen. So one of these events is that translation decreases to reduce the folding load of the ER. And this would make sense because if you have problems with proteins in the ER, the cell wouldn't want to start producing new proteins. So it just would want to focus on the problem at hand before making even more proteins. Another thing that occurs in the UPR is that the chaperone production increases. And this would make sense also because you have proteins that are behaving irregularly. So you would need more chaperone proteins to be around to help the unfolded proteins refold correctly. And the third thing that happens in this response is the lipid synthesis increases. And this will be helpful to the ER because it will maximize the volume or the space inside the ER. So it can accommodate for the increased load of proteins that are inside the ER. And so for this part, how does the ER get rid of misfolded proteins? So if the protein cannot be refolded correctly, if the chaperone proteins are unable to refold the protein correctly, then it has to leave the ER. And since there are no proteasomes in the ER, the protein has to be retrotranslocated, which means that it has to be removed from the ER so that it can be sent to the proteasome. And yeah, that's pretty much it for the UPR. Does anybody have any questions on this slide here? Okay. And yeah, this is just a diagram I thought I'd include. Don't get overwhelmed by all these proteins in here. She's not expecting you to know the names of all of these things, but this is just a visual that shows what's happening in the ER. So you have like upregulation of chaperones and then lipid synthesis, and then you have reduced translation and some of the other things I talked about in the slide. So this is just a visual of that for people who like to look at things visually. Okay, so I'll just keep going. And Jason, do you want to do this next part?
Sure. All right, so now we'll talk about protein ligand interactions pretty quickly. So if you don't know, a ligand is just anything that a protein can bind to. We use that as a broad umbrella term. Usually the ligand involves a binding pocket, which we'll show on the next slide, but it doesn't always have to. It could also bind to like a flat surface on the cell. Within the binding pocket or however the protein binds to the ligand, the protein needs to be oriented correctly. And how it does its reaction is the amino acids need to be exposed from the pocket. So remember the R group interactions we talked about, these play a role on how the proteins can bind in the binding pocket. And for example, a protein with like really large amino acid groups, like those benzene rings that Rachel was talking about, those might not always fit in every binding pocket. So remember, one of the biggest things from this class is that structure influences function. So the structure of the protein could have an interaction on what things it can bind to. Next slide. So affinity is just the strength of the interaction between the protein and whatever it's binding to. So this is the binding pocket that I was talking about a second ago. Affinity is dependent on two main things: the number of bonds that are formed between the protein and the ligand, and the strength of the bonds that are formed. So if there's more bonds, or if there's, for example, like a covalent bond, that'll be much higher affinity. You rarely have a covalent bond between a protein and a ligand because that's super hard to break, but just an example of something that would give a higher affinity. Laura didn't really talk about this, but two things to also consider are the type of the bond and the angle of the bond. So the thing we talked about, the concept we talked about in relation to affinity is the dissociation constant. And this is just a way to measure and describe protein-ligand interactions. So it's the average rate measurement of how often the protein dissociates from the ligand, and it's the concentration of the ligand when half the binding sites in the protein are filled, or when the protein concentration equals the complex concentration. Don't get too bogged down in the formula, so we'll never ask you to like calculate a formula. But the biggest thing here is that there's an inverse relationship between KD and affinity. So as affinity increases, KD will go down. So a protein that makes really strong bonds will have a low KD. In a protein that makes really weak bonds will have a high KD. So that's the biggest thing there. She might ask a few questions in relation to that. Next slide. So one way to help conceptualize KD is to think about enzymes. So remember the Goldilocks hypothesis that Michelle was talking about earlier? You don't always want to have really strong bonds. Sometimes you want the protein to let go of whatever it's binding to. So for example, you want an enzyme to bind strongly to a substrate or the reactants. And so the KD would be lower here because we want the enzyme to be securely attached to the substrate. But once the product is formed, the enzyme needs to leave so it can go do something else and let the protein do its thing. So you'd want the KD to be higher here so the enzyme can leave now that the product and the reaction is finished. If the enzyme gets stuck because it has too low of a KD, then you wouldn't be able to use it again and you wouldn't be able to recycle the enzyme and it would cause lots of problems in the cell. So that's everything I have for KD stuff. Does anyone have any questions here before I move on to enzymes in general? Great.
All right, so we'll go through enzymes. I know we didn't spend a ton of time on enzymes in class, but just brief overview like we've been saying for most of this. Remember that this is also 25% of everything you need to really kind of know for the final. So anyways, enzymes, they are catalysts that carry out chemical reactions. They make and break covalent bonds. The formation and destruction of covalent bonds are chemical reactions. Just an example of the enzyme that catalyzes the breaking of that bond is called a peptidase. It breaks apart those peptide bonds. Ribosomes form covalent bonds or peptide bonds between amino acids, and spliceosomes are an example of enzymes that break covalent bonds between sugar molecules. So they're important to cells because they change the chemical structure of molecules, and they can also be easily regulated, which is important because this means that cellular processes that rely on enzymes can also be regulated. So the cell has a way of, controlling what it doesn't break, puts together, all that stuff. So another important point to know is that not all enzymes are made of 100% protein. There are some called ribozymes that are made of an RNA protein mixture, in which RNA is the catalytic molecule. So they are usually made of protein, but not always. If she ever asks you about it, which I doubt she will, that just means that it's RNA and protein. Okay, so to continue with enzymes, they have to bind to a substrate for catalysis to occur. So they have to have some affinity for the substrate. Enzymes can undergo conformational shifts during a reaction, but they come out unchanged at the end of the reaction. So when they're bound to the substrate, you can see in these two diagrams, they usually change conformation either to have more affinity to bind the substrate, and but it will come out of the reaction the same way it went in. So it's important for enzymes to have a higher affinity for reactants than products, because we want the enzymes to latch onto the substrate, which is the reactant. Then they will catalyze the reaction and dissociate from the product. We want to reuse the enzyme after the reaction, so that's why it reuse it, so it comes out the same, so we can reuse it in the cell. And then, like Jason mentioned before, higher affinity is the same as having a low KD. Yeah, that's all I have to say on that slide, actually. So this is why enzymes are useful. They're useful in chemical reactions. They can speed up reactions that take too long if they occur on their own or spontaneously. It increases the frequency of chemical reactions. It can pull two substrates close together or bring them closer to make a reaction occur that would probably take too long. And they can change conformation of the substrates by straining them to make a reaction favor the transition state of the reaction. They are also useful for cellular function. So the time for the spontaneous reaction without an enzyme is usually much too slow for the cell to function properly. I know we mentioned with, RAN gap, that without it, the cell would be too slow to dissociate, or with RAN-GAF, and it would be so slow that it was almost like it didn't even have the function of doing the reaction on their own. So the enzymes help with that. They're regulated, like I said before, so that if the enzymes can be regulated, then the cellular processes that the enzymes are a part of can also be regulated. And this is important for overall cell function. So why is it beneficial for the enzyme to lower the activation energy of the reaction? Reactions occur more frequently and it's more energy efficient. So without the enzyme, you can see on this graph, that's that green line, the activation energy is much higher. That's what EA stands for. But when you add the enzyme which is this pink line, the activation energy is much lower. It's easier to get over this hump, and the reaction will occur more favorably. Just another quick note, more stable in this case with enzymes is less reactive and less energy. So the less stable you are, the more energy you have. The more stable you are, the less energy you have. So here we have these diagrams. I think they're from your textbook. I know she showed them in class before, so these are the three ways that the enzymes work by binding to the substrates. So A is showing the enzyme bringing two substrates closer together, so it orients them to allow a reaction to occur. So maybe these two substrates were, flying around the cell, and the odds of them coming in contact were pretty low, but the enzyme orients them in the way that they're needed for a reaction to occur. B shows that the binding of a substrate to enzymes rearranges electrons in the substrate, which creates this partial negative and partial positive charges to favor a reaction. And then C shows the enzyme straining the bound substrate to force it into a favorable transition state for the reaction to occur. So yeah, that's, I think I'll just say about that one too. So wait, this is still me, right?
Yep.
Okay. Hold on, let me just catch up on my slide. Okay, so post-translational modifications, they occur after the protein is made. So this is after tertiary structure or quaternary structure, depending on your protein. The functional group is added covalently and regulated by an enzyme. So it's the functional group that's added is regulated by an enzyme, and it can also be removed by an enzyme as well. And basically what the effect of post-translational modifications is to change the protein surface chemistry or its structure and its function. So in this diagram, I think you guys have seen this before, this yellow here is adding on phosphorylation. This green at the bottom is ubiquitin, and blue is acetylation. So basically what happens is the cell signals direct the addition of covalent modifications. So here you have your protein here, your N-terminus, and your C-terminus at the end. the code is red, and it's either going to bind to proteins Y and Z, or move to the nucleus, or move to the proteasome for degradation, or move to the plasma membrane. So based on the post-translational modifications, it will depend on which one of these four outcomes the protein will have based on what's added. So the main thing to take away from that is just that form follows function. That's just structure always equals function. Does anyone have any questions so far?
Yeah, I was just gonna add that. Sorry, I feel like I'm echoing, but I was just gonna add that form follows function, and then depending on what functional group is added, you can change the function of a protein. So like a ubiquitin might have a different function than a phosphate group, and so that's why there's like this. That's why after the protein is made, these groups can be added, and it can give proteins different jobs to do in the cell.
Yeah. Another example too is P53. Now you guys know more about that. Is an example of a protein that's post-translationally modified in multiple ways. You don't need to know like what ways it is, but it's just one of those proteins that is.
Yeah, and P53 I think is involved in DNA damage, but you guys will hear more about that in the cell cycle review. Yeah. All right, so I'll just keep going then. And oh, sorry, Rachel.
No, it's fine.
Okay. So then this is nucleotide binding / hydrolysis, and it can lead to different binding and active states. And then proteins that are GTPases and ATPases are enzymes. So these proteins are enzymes, and they can bind to GTP or ATP. And these particular proteins have the ability to change or cleave bonds between phosphates and change GTP, change GTP to GDP, and in the opposite direction as well. So then it would be ATP to ADP and vice versa. And then it's a regulatory mechanism, and it's common in pathways. And we're going to see this once we go through the pathways, which I think is tomorrow's review. But anyways, I tend to think of the nucleotide binding of GDP and GTP as light switches, because when they're bound to proteins, they can either activate or deactivate them. And so GTP, as you can see at the bottom, is activating the protein. So the protein is in its active on state. And then once the extra phosphate is cleaved, becomes GDP, which is the protein in its inactive state. And yeah, so that's nucleotide binding, and you'll definitely see that in the nuclear import, nuclear export, and a lot of the other pathways as mechanisms to activate or deactivate proteins. So does anybody have any questions on this slide? Okay. And now we're going to jump into protein localization or protein trafficking. And oh goodness, the slide is, yeah, it's okay. I'm gonna read what's at the bottom there, but I think it's been cut off. But anyways, so in eukaryotic cells, proteins are made in the cytosol. And within the cytosol, you have ribosomes, and there are two types of ribosomes. There are ribosomes that can float freely in the cytosol, and then there are, I believe, membrane-bound ribosomes that are attached to the side of membranes on the rough ER. And proteins that go in the endomembrane system are made by ribosomes that latch onto the outside of the ER membrane during translation. So those are ribosomes that are docked to the rough ER. And proteins that are heading into the endomembrane system go to the ER, the lysosome, Golgi body, or they can get secreted all the way to the outside of the cell. And proteins that don't go to the endomembrane system will be made on ribosomes that float freely in the cytosol. And these proteins are cytosolic proteins, nuclear proteins, mitochondrial proteins, and I think chloroplast proteins, but we won't be talking about chloroplast proteins in this class. So does anybody have any questions on this slide? Okay. And yeah, so this is just a visual of what I just said. So you have the free ribosomes in the cytosol, and then you have membrane-bound ribosomes, which are directly attached to the membrane of the ER. And yeah, this is just a diagram of everything I said. And I'm gonna keep going.
I have one question.
Yep.
So for the endomembrane system, the ribosomes can either be bound, like membrane bound, or they'll be secreted.
Can you repeat your question? I just want to make sure I heard it correctly.
Sorry. So if something's in the endomembrane system, they're either secreted or they're membrane bound, or is it that if they go into the endomembrane system, they're automatically secreted?
So for the endomembrane system, it's not that they're automatically secreted. It just depends on what signal that particular protein can have. But for the protein to be able to enter the endomembrane system, it has to start with the ER. And so for the protein to get into the ER, it has to be translated on a ribosome, and a particular ribosome, and that particular ribosome is the ribosome that is membrane bound. So if the protein is going to go into the endomembrane system, and let's say it was going to go and end up on the outside of the cell, it can't be translated on a freely floating ribosome in the cytosol. Otherwise, it will never get there, because it has to be attached to the membrane-bound ribosome because of the mechanism of how those proteins are made. They sort of just get funneled into the ER while it's being synthesized. So that's why those proteins have to come from the membrane-bound ribosomes. So does that make sense?
Yeah, thank you.
Yeah, and if it still is kind of fuzzy, I'm gonna go through like the difference between how the translation occurs for those proteins that go outside versus the ones that go to the nucleus and other places. Also, I see messages in the chat, like in my screen, but I can't read them. I just see the notification that says I have 15 chats. So if one of the TAs could look at the chat and let me know if there are questions in there.
Someone just said, I think secretion is the fault of the endomembrane system if the protein does not have another retention localization sequence. Is that correct?
That is correct, but I'm going to get to that once I keep going through the slides, and it should make more sense. But the default of the endomembrane system is that if there's no other signal, so if it had a signal sequence that said the protein was going into the ER, but then it had nothing else after that, that protein is eventually going to end up being secreted outside of the cell, because for it to stay within the ER, it needs an ER localization sequence and then an ER retention sequence. But again, I'm going to get to all of this in the next slides, so it will make a lot more sense once we get there. But that is correct. Okay. So now I'm going to talk about the endomembrane system, and I'm so sorry, but it looks like the bottom of the slide is hard to read, but I'm going to make sure I cover the information on there. So endomembrane system is just a group of membrane-bound organelles that work together to transport proteins. And like I said, it's just composed of components such as the ER, Golgi bodies, lysosome, cell membrane, and transport vesicles that move between them. And trafficking of these proteins through this particular system is called the secretory pathway. And so any protein that you have that functions in the ER, Golgi body, lysosome, transmembrane protein, or a protein that's being secreted to the outside of the cell has to travel through the endomembrane system. And movement of proteins through the endomembrane system has to start with the ER. And so the way that the general overview of that is just the membrane-bound vesicles can taper off of the ER and move to the Golgi bodies. And then the vesicles, then the protein will go through the Golgi body, and then it will just pop off the vesicle of the Golgi and go to either the lysosome, cell membrane, or even outside of the cell. And so just like we just mentioned, but by default, if the protein only has ER localization sequence, it will continue to travel until it gets secreted out of the cell. And it won't stop unless it has a retention sequence somewhere, or if it has another signal. Does anybody have any questions on this slide? Okay, I'll keep going. And so, yeah, this is just a diagram, I think, either from your textbook or something of the endomembrane system. So it's just like I said, everything begins in the ER, which is at the bottom of this screen. And so if the protein is heading to the outside of the cell, it goes through the ER, then it pops off of the vesicles, then the vesicle will attach to the Golgi bodies, it goes through the Golgi, and so on and so forth until that protein reaches the outside of the cell. So yeah, I just thought this was a nice visual to include in there. And now I'm going to talk about signal sequences. And so signal sequences are embedded in the amino acid sequence of a protein. And it's something that's not added after the protein is being made, which is what distinguishes these from post-translational modifications. I just thought that would be important to emphasize because it's not added after the protein is being made, it's just within its sequence. So signal sequences are basically like a zip code. So it tells the protein where its destination will be or where it will be living, sort of. And so you just have to remember that every time a protein is localized to the ER, it has to have an ER signal sequence. And this is due to the mechanism of ER trafficking because the ER signal sequence is recognized by SRP, which is a signal recognition particle, and it will bind to the newly made protein and take it to the ER. But if a protein does not have ER signal sequence, then it's unable to leave the cytosol. So it will stay in the cytosol after it's been translated. And then if it has a different sequence that's not ER, then it will follow that particular sequence. And just like I mentioned before, any protein that has to go outside of the cell has to go through the endomembrane system. And once proteins leave the ER, they generally never come back out again. But they never, once a protein leaves the ER, they usually don't return to the ER. But the process by which they return is called ERAD, but this is a very rare process, and I don't think she talked about that in the class, for this class. Does anyone have questions on this slide? Okay, I'm just gonna keep going. And so this was a diagram I saw on Discord, very helpful. I had this diagram for Bio 151, but it's just you can run through it and just see how to figure out where a protein will end up based on what sequence it has. So you start with the top portion, so you ask yourself, does it have an ER signal sequence? And then if it does, then it will end up being translated by a membrane-bound ribosome. And then you sort of follow the schematic here and figure out where the protein would go. In this class, we did not cover signal anchor sequence, but signal anchor sequence is a special sequence for a transmembrane protein, but you don't have to know that for this class. So I don't think she will ask you what a signal anchor sequence is, but it's just included in this diagram. So if you wanted to figure out if a protein went outside the cell, you would just sort of go through the schematic and you would just say that the ER signal sequence, yes, it has that, doesn't have an anchor sequence. And then does it have other signals? No. And then if it's not embedded in the membrane, then it gets secreted. And so that's just a diagram that does that.
She doesn't talk about signal anchor sequences, but she doesn't talk about something like an ER retention sequence. So if something needs to stay in the ER, you would need like an ER retention sequence, which isn't in this diagram, but a term that she does use.
Yeah. So for a protein to stay and work in the ER, it needs to have an ER signal sequence and then ER retention sequence. But the diagram does not show the retention sequence. But I just thought this diagram was a good idea for just overview of looking at where proteins go. But just know that you need 2 sequences for a protein to stay in the ER.
I have a question.
Yep.
So would that ER retention sequence, would that maybe be at the bottom of the diagram where it says goes back to ER? If you were to put it somewhere on this?
I guess that's probably what that would be. That would make sense.
I don't think it'd be at the bottom. I think it would be for ER retention, it would be before the protein is transported to Golgi. Is that what you're talking about?
Yeah, for this diagram, I just feel like it's slightly different than what you learned because you have to have the retention sequence. But I feel like when it says protein enters ER during translation, they should ask a question maybe before the anchor sequence, like does it have a retention? Then maybe that's where it goes. So yeah, I think Rachel's idea of it is, I think, better. So I don't think it would go at the very end, if that makes sense.
Okay, thank you.
Yeah. But yeah, now I'm looking at this diagram, I'm just thinking it's more confusing and useful. But yeah, I just for this class, just focus on the content that you've learned. Don't get confused by the diagram. If the diagram's helpful, you can use it. But if it just makes you more confused, then I wouldn't use it. But yeah, I'm just gonna continue. Okay, so more on signal sequences. Signal sequences target proteins to their final destination, which I said. Professor Francis is big on saying that signal sequences act as binding sites for another protein. And a great example of this that you've seen before in the nuclear import and export is how Importin, which is a special protein that's involved in nuclear import, searches for the NLS on the exterior of a protein that's entering the nucleus. So in that sense, the nuclear localization sequence, or the NLS, acts as a binding site for the important protein to bring the protein that's destined to go into the nucleus. Okay, and then typically proteins will enter the organelles unfolded. So what I mean by this is that proteins will enter organelles sort of unraveled. And we've seen this before when proteins go into the ER, but the only exception to this rule is proteins that get localized to the nucleus. So when a protein has been made, fully created, so when it's made in tertiary structure or quaternary structure, depending on the protein will stay in that shape. So it's not going to unravel itself while it enters the nucleus. And this is because the nucleus has nuclear pores, and these are able to accommodate for proteins that are entering there, so they can expand and contract to let proteins in through the pores. So the proteins that are going into the nucleus are fully folded and fully made, so they'll squeeze through the nuclear pores, and that process is regulated. And N-terminal sequences are usually cleaved before, N-terminal sequences are usually cleaved before proteins go into the ER in mitochondria, so it doesn't, I'm actually thinking the typo here. So yeah, once the protein goes into the ER or the mitochondria, the N-terminal sequence is cleaved so that it does not exit the organelle. I think that's what that's supposed to say. Rachel or Jason, can you make sure, can you check to see that I'm saying that right? Because I feel like I might have said something wrong. I believe that it is, it's cleaved. I just feel like I worded that wrong.
Yeah, it gets cleaved like right when it enters the organelle, so it can't go back out.
Yeah. Yeah, sorry. Yeah, so the N-terminal sequence gets cleaved, so it does not exit the organelle. Does anybody have questions on this slide? Okay, I'm gonna continue, but I have a summary for each one because they're difficult to keep track of. And so yeah, this is just another visual of protein localization. And so it just shows how you have proteins that, every single protein that you see here, that's going to a different destination, they all start off being made in the cytosol. And then depending on what kind of ribosome it's on, dictates where it will end up going. And so yeah, that's just the diagram. Does anyone have questions on this diagram? And yeah, this is just a summary of everything, just because I feel like it can be difficult to keep track of sometimes. And so you can see that for the nucleus, the signal sequence will remain on the protein all the time, so it doesn't get cleaved. And then for the ER and mitochondria, I'm sorry, that's what I was supposed to say. The signal sequence will be cleaved by a protein called a signal peptidase after that protein enters the organelle. And so that's to prevent it from getting back out again, from leaving the organelle. So that's true for the ER and the mitochondria. And then for the nucleus, the signal sequence will be found on the surface of the folded protein, whereas for the ER and mitochondria, the signal sequence is embedded into the amino acid sequence, which is why I have it phrased as the signal sequence is embedded into the primary structure. So it's not something that's added after the fact in either of those organelles. Signal sequence is something that's already part of the protein itself. It's not an extra addition to a protein. And for the nucleus, the protein can enter fully folded and fully made because it can squeeze through the nuclear pores. Whereas for the ER and mitochondria, the difference here is that the protein will actually unravel itself and it will enter the organelle sort of uncoiled, in an uncoiled state. And then once it gets inside the organelle, the signal sequence is cleaved, and then there are chaperone proteins in there to help the protein refold into its proper structure. And for ER and mitochondria, the signal sequence is located in the N-terminus of the amino acid sequence. And then, just like I said, signal sequence is on the outer surface of the protein for the nucleus, for the nuclear proteins. So this is just a summary of what I said. I find this one a lot easier to read, I feel like, than the others. But does anybody have any questions on this slide? No? Okay. So this is just a diagram of kind of reiterating what I was saying earlier, and how you see how the ribosome docks to the ER, and then while the protein is being synthesized by the ribosome, the sequence is recognized by SRP, and then it draws it into the ER, and then it draws the protein into the ER, and then the signal, which is on the N-terminus, gets cleaved by signal peptidase. And you can see that the protein that goes into the ER is entering that organelle in an unfolded state, so it's sort of unraveled, kind of like spaghetti, and then the chaperone protein will assist in folding it correctly, so it becomes folded while it's inside the ER. And it's similar in the case of the mitochondrial proteins that end up in a mitochondria. And so there's recognition of the sequence, and then it gets taken to the mitochondria and it enters unfolded. Then the signal is cleaved, and then it's refolded into its proper form. Does anyone have any questions on this slide? Okay, I'm gonna keep going. Okay, and so this is just a diagram of a variety of signal sequences, and they're just the important ones I thought that were good to know for this class. She talks a little bit about peroxisomes and stuff like that, but I think in general you should just focus on the ER, mitochondria, and nucleus, because I think that's where most of her questions come from these types of proteins. And as you can see, something that's important to know is that the cytosol does not have any signal sequences. So if you were asked a question where she removed signal sequences and there was no signal sequences, and you had to figure out where the protein would end up, just to know that it would most likely end up in the cytosol. Or another question that she liked to ask, at least when I took the course, was what would happen if you had a protein that just had an ER retention sequence. And so there was a misconception that a lot of people thought that the protein would end up in the ER, but that it would actually end up in the cytosol, because if you just have an ER retention sequence and no ER import sequence, then that protein will never be localized to the ER. So that sequence of retention would just sort of be useless. So it would end up in the cytosol. And other things to highlight is that the signal sequences can vary in chemistry. So for mitochondria, the signal sequences are just interspersed positively charged R groups on the N-terminus. And then for the ER, you have hydrophobic amino acids followed by interspersed charged R groups. And so this is why, since the signal sequences have varying types of chemistry, this is why when you tweak amino acids in a signal sequence, you can have really strong effects on a protein. So if you changed a couple of amino acids in the NLS, then you may prevent the important protein from recognizing the NLS, and it won't be able to bring that protein into the nucleus because each type of signal sequence, depending on where the protein is going, it has its own specific chemistry that it's supposed to follow. So it's important that those signals remain the way that they are, signal sequences. Does anybody have any questions about this slide? Okay. And yeah, so this picture I think is a little bit better than the other one. So it just sort of shows how there's different types of signal sequences, like I was saying before, and that they differ in their chemistry. And yeah, so that's just pretty much what I was saying before. And if you change even just a few amino acids in one of them, then you can really have harmful effects on the protein, and you could cause a protein to not go where it's supposed to go. So does anybody have any questions on this slide? Okay. And so I actually have a question here. So transmembrane proteins must have ER import signal sequences on their N-terminus. And so predict where you would find a transmembrane protein if you engineered its gene such that you removed the ER signal sequence. And assuming that your receptor protein does not completely misfold or get degraded, where will your receptor end up? So if anybody wants to put it in the chat or say it out loud, I can't see the chat. So if somebody wants to read it to me.
Cytosol?
Yeah, so this would actually end up in the cytosol. And this is a tricky question, but this is actually one she asked me when I took this course. So I just figured it'd be good to go through it. So yeah, the answer is cytosol. And so you'd expect to find the protein in the cytosol. And this is because the receptor needs the ER signal sequences for the SRP to recruit it. And then it will be able to become stuck in the ER membrane. And let me see. The receptor needs the ER signal sequence for SRP to recruit it to the translocation channel, and then it will become stuck in the ER membrane and then be sent out to the cell membrane. A portion of the protein will stick out to the cell surface while the other portion is stuck inside the cell. So without the ER signal or any other kinds of signal sequences, the receptor would just end up floating in the cytosol. And so I just have an analogy for this in real life. So it's just sort of like if you were getting an Uber, and if you don't tell the driver your location, then they won't find you and transport you to your destination. So it sort of works similarly with the signal sequence. So if there's no ER signal sequence on a newly synthesized protein, then there's no way for the SRP, the signal recognition particle, to recognize the protein and bring it to its final destination, the ER. So does anybody have any questions on that example question? Does it make sense why it would end up in the cytosol? I just want to make sure it makes sense. So if someone can just be like, yeah, it makes sense, then I feel comfortable moving.
Yeah, it makes sense.
Okay.
Yeah.
Okay. And so example of protein trafficking, or I guess just trafficking in general in real life, is hormone secretion. And so depending on hormone's final destination, it can determine if other signal sequences are attached to it. And so hormones, I don't think hormones are protein. So it's just an example of sorting and trafficking. And you can see how the endomembrane system is being used here. You have the ER, the Golgi, and the vesicles. And yeah, so the hormone just goes through the endomembrane system. But I think the hormone is a protein here because it seems to be being translated by the ribosome. So yeah, hormone is a protein in this one. Okay, does anyone have any questions on this slide? Okay. And yeah, I think this is our last section for today.
Yep, so last thing we're going to talk about is nuclear import and export. So the first thing you need to go over to understand nuclear import and export are GEFs and GAPs. Also, just a clarifying thing, a lot of people had confusion on the last exam. Laura put a question about like that included what a gap was on the exam. People thought that we never covered it before, but this is where we covered it. She mentioned RAN gap, but GEFs and GAPs are proteins that regulate the whole family of GTPases. RAN and RAS are both part of the same family of GTPases. So either case, they have the same general function. So to understand GEFs and GAPs, I think it's most helpful to look at the names of the proteins. So GEF is guanine nucleotide exchange factor, and it does pretty much what the name describes. So basically what it does is it facilitates the exchange of GDP to GDP. And basically you can think of it as kind of like a shovel. So what it does is it kind of removes the GDP from the binding pocket. GTP is present in higher concentrations outside of the binding pocket, so it'll diffuse into the binding pocket once GDP is removed. So the end result is that it makes the GTPase active, so it can does its job in the nucleus, and result is that RAN is bound to GTP, sorry. The other protein is a GAP, and GAP, again, look at the name, it's a GTPase activating protein. So what it does is it basically hydrolyzes GTP to GDP. So RAS can do that by itself, but basically it kind of acts as a sort of enzyme to make it go faster. So the end result is it makes RAS inactive, so it can do its job in the cytosol. And the end result is RAN is bound to GDP.
Yeah, I also just wanted to real quick just interject and explain the gap real quickly, because Professor Francis wants people to use the correct terminology. So when you talk about the pathways and you say that gap hydrolyzes GTP to GDP, that wouldn't be the correct terminology, because proteins like RAN have their own intrinsic GTPase activity. So RAN is an enzyme, and enzymes can catalyze reactions. So RAN itself is the one that's doing the hydrolyzing of GTP to GDP, but it does it much slower, and it takes a while. So what it does, it recruits the help of GAP, and GAP sort of accelerates the hydrolysis. So GAP itself is not hydrolyzing the GTP, it's just quickening RAN's ability to do so. So I hope that makes sense, but it's just sort of, it would just be wrong to say that GAP is hydrolyzing GTP, when in reality it's RAN or whatever GTPase you're dealing with in the pathway is doing that itself.
Great, so next slide. We're gonna go over the slide really quickly because I just explained this, but basically GAPs bind to RAN GDP and cause a change in conformation so that RAN no longer has a change in GDP, good affinity for GDP. So basically all it does is just switches out the GDP for GTP. GAP is the opposite. GAP allows for it accelerates the hydrolysis of GTP to GDP in the RAN's binding pocket. So GAP will turn, well, basically as a result of GAP will result in RAN GTP becoming RAN GDP. And now we can talk about how these proteins function in nuclear import and export. So stop me if you have any questions, but basically I'm just going to go over the whole process. So first, for nuclear import, a protein that has an NLS or a nuclear localization sequence, it needs the NLS to be able to bind to important. If it doesn't have it, it can't bind important. So proteins with an NLS can bind to important. They enter the nucleus through the nuclear pore complex. Remember that proteins can enter the nucleus fully folded, so it doesn't have to unfold to enter the nucleus. It can just enter the nucleus fully folded. Once it's in the nucleus, it needs to release the cargo protein that it just brought in. So it does that by when the GTP on RAN, RAN will bind to the important cargo protein complex, and then once it's switched to GDP, it will allow the cargo protein to be released. So now the RAN GTP complex needs to exit the nucleus. So it does that again, fully folded through the nuclear pore complex. And so now we need to release important from the RAN GTP complex. This does that with the help of GAP. GAP accelerates the intrinsic GTPase activity of RAN. So as a result, RAN GTP will become RAN GDP, and in this process, important will be released into the cytosol, so it can redo the process again. Does anybody have any questions on this? I know a lot of people struggled with the question on the stuff in the first exam. So like she asked about like, what if RAN, what if GEF could function in the nucleus and the cytosol? Basically, if GEF was also in the cytosol, what would happen is it would just keep causing the RAN complex to bind important again because it would be bound to GDP, and it wouldn't be able to keep going through its process, and nuclear import would stall. So, stuff like that. So that's the kind of question she's going to ask. Probably something similar. I think she asked about nuclear export on the exam, so she'd probably ask about like nuclear import on the final, something like that. Anybody have any questions on this? If not, we can talk about nuclear export.
Yeah, and I think she might actually, I'm not too sure on this, but she might have like, I don't know if you guys see, but she posts like practice questions for each week. So there might be practice questions on the types, which demonstrate the types of questions she would ask on the final on nuclear import and export. So I'd recommend going through those and seeing if you can understand those.
Oh, also, a lot of people messed up. A protein that lives in the nucleus would need an NLS, right? But to get out of the nucleus, you'd need to add a nuclear export sequence. Okay. If a protein is just functioning in the cytosol, you would not have any signal sequence originally. So a GAP would not originally have any signal sequence, but if you needed GEF to function in the nucleus and the cytosol, GEF is normally functioning in the nucleus. So to get it into the cytosol, you need to add a nuclear export sequence. Okay. Just extra stuff from the first exam. So nuclear export is just the opposite of nuclear import. So you have something in the nucleus, you want to get it out of the nucleus. So basically what happens is you have this RAN, GTP exportin complex, Okay, yes. So you have RAN GDP in the nucleus. Once GEF activates it, it switches GDP to GDP, and now the RAN GTP complex can bind exportin. Once it's bound to exportin, exportin will bind the protein that needs to go out of the nucleus. If it needs to go out of the nucleus, it needs to have this NES. So it exits the nucleus, and then Once it's in the cytosol, you need to release the cargo protein into the cytosol. So GAP has to help accelerate the intrinsic GTPase activity of RAN. So it'll allow it to hydrolyze GTP to GDP much faster. Once it's hydrolyzed to GDP, the exportin is released and the cargo protein is also released, and then the nuclear export cycle can continue. Anybody have any questions about nuclear import and export or anything we covered throughout this whole exam review? Any exam one review content? Otherwise, we're pretty much done.